In plants, the products of meiosis divide mitotically to form haploid gametophytes. The gametophyte phase of a plant life cycle alternates with the sporophyte phase, which is diploid meiosis occurs in the sporophyte. Key points. Diploid eukaryotic cells form haploid cells by meiosis, a process involving one round of chromosomes duplication followed by two cell divisions, meiosis 1 and meiosis 2. During meiosis 1, homologous chromosomes pair synapse, exchange material crossover, and separate disjoint from each other. During meiosis 2, chromatids disjoint from each other. In many organisms, the haploid products of meiosis develop directly in the gametes. The formation of gametes, a process called gametogenesis, occurs in the gonads of animals. Oogenesis, the formation of eggs, occurs in the ovaries, which are the female gonad, and spermatogenesis, the formation of sperm, occurs in the testes, which are the male gonads. These processes begin when undifferentiated diploid cells called oogonia or spermatoogonia undergo meiosis to produce haploid cells. The haploid cells then differentiate into mature gametes. The existence of both haploid and diploid organisms in the life cycle of plants is referred to as alternation of generations. Among animals, the haploid products of meiosis develop directly in the gametes. Usually only one of 4 haploid cells from female meiosis become an egg, or ovum. The other 3 cells, called polar bodies, degenerate. By contrast, all 4 of the haploid cells from male meiosis develop in the sperm. Only one of the 4 haploid cells produced by female meiosis develops in the gametophytes. This is called the megaspore. All 4 of the haploid cells from male meiosis, called microspores, develop into gametophytes. When mature, these haploid plants produce gametes, either eggs or sperm. Consequently, they are called gametophytes. The gametes unite at fertilization to form diploid zygotes, which divide mitotically and develop into structures called sporophytes. In higher plants, the gametophytes is much reduced in size. It consists of just a few haploid cells. Thus, the sporophyte is a conspicuous part of life cycle. Meiosis occurs in distinct male and female reproductive tissues in the sporophytes of higher plants. Nucleus is formed when the other sperm cell nucleus combines with the diploid secondary endosperm nucleus of the female gametophyte. When a mature pollen grain lands on the stigma's top, the pistil and pollen tube grows down through the style to an egg cell within the ovary. In plants, fertilization involves 2 events. One, the diploid zygote, which will grow into an embryo, is formed when one sperm cell from the pollen tube fuses with the egg cell of the female gametophyte, the triploid endosperm. Cytokinesis then occurs, creating 3 antipodal cells, 2 synergid cells and an egg cell. 2 polar nuclei remain in a large central cell of the embryo sac. These polar nuclei subsequently fuse to form diploid secondary endospore nucleus, and the 3 antipodal cells degenerate. The haploid nucleus in each megaspore undergoes 3 mitotic divisions to produce an immature embryo sac containing 8 haploid nuclei. Meiosis in the female tissue produces 4 cells. However, 3 subsequently degenerate, leaving only 1 functional megaspore. Female gametes, or microspores, are produced from megaspore mother cells by meiosis in the ovary, which is located within the pistil at the center of the flower. Each microspore then undergoes mitosis to produce a mature pollen grain, which contains 2 generative or sperm cells located within a vegetative cell. Each of these cells contains a haploid nucleus. Arabidopsis is small and it has a relatively short generation time, about 5 weeks. Like Mendel's garden peas, Arabidopsis is a self-fertilizing species. However, different strains can be cross-fertilized in the laboratory to produce hybrids. The male gametes, or microspores, are produced from microspore mother cells by meiosis and antlers, which are atop the stamens within Arabidopsis flowers. Garden plants were the first organisms to be studied genetically. Today, geneticists focus their attention on Arabidopsis thaliana, a weed sometimes called the mouse ear cress. This member of the crucifer family is related to food plants such as radish, cabbage, and canola. However, it has no agronomic or horticultural value. Today's Dr. Will, CBMD, PhD, today's Thursday, February 8, 2018. Continuation. Arabidopsis thaliana, a fast-growing plant, by capturing solar energy and storing it in simple sugars such as glucose, plants play an indispensable role in the web of life. As a byproduct of this process, they also generate oxygen, which and all other non-photosynthetic organisms breathe. However, the number of genes in multicellular animals is only 2 to 3 times greater than the number of genes in yeast. Geneticists are trying to reconcile this relatively modest increase in gene number with the dramatic increase in biological complexity. Both the Drosophila and C. elegans genomes have been sequenced. These genomes are much larger than the yeast genome, 100 times 10 to the power 6 nucleotide pairs for C. elegans and 170 times 10 to the power 6 nucleotide pairs for Drosophila, and they contain more genes, an estimated 20,516 in C. elegans and 13,792 in Drosophila. Another invertebrate to nematode Cenohabditis elegans, known to geneticists simply as C. elegans, also has played a major role in the genetic analysis and development. Thousands of mutant strains of Drosophila have been obtained in the nearly 1,000 years this animal has been studied. Some show unusual eye or body colors, others have an abnormal anatomy, short wings, extra legs, misplaced bristles, and so on. In one strain, the halters are converted into wings. The anatomical complexity of Drosophila provides geneticists with opportunities to study how genes control the formation of body parts. It also allows them to investigate the processes by which a fertilized egg develops into an embryo and eventually into an adult. The third larva instar are the last stage before metamorphosis into omega, or adult fly. The skin of the third instar larva hardens into a case, and the animal becomes a pupa, plural pupa, a stage during which the larva's tissue are radically recognized. Packets of cells called imaginal discs grow and differentiate into adult structures such as eyes, wings, and legs. The larva feeds voraciously for about a week. At intervals during this period it sheds its skin to allow for increase in body size. Each larva stage between these molts is called an instar. A female Drosophila can produce hundreds of eggs. When fertilized, each egg becomes an embryo, which hatches out of an eggshell to become a worm-like larva, plural larvae. The surface of the adult body is covered with sensory hairs and bristles, which are connected to the nervous system. Other prominent sensory organs, the eyes and the antennae, are located on the head. The reproductive organs are located in the abdomen. The adult fly is about 2 millimeters long and possesses a complex nervous system and many specialized tissues and organs. Like all insects, an adult Drosophila has 3 pairs of legs. However, unlike most insects, it has only 1 pair of wings. The second pair of wings has been modified into small appendages called halters, which help the animal to balance during flight. The fruit fly Drosophila melanogaster came on the genetic scene in 1909, decades before bacteria and fungi. It appealed to researchers because it is easy to rear in the laboratory. It has a relatively short life cycle, about 10 days and 25 degrees Celsius. Drosophila melanogaster, a fruit fly. With untold millions of species, insects are the most diverse group of animals on Earth. It therefore seems appropriate that one of the model organisms for genetic analysis is an insect. The 4 haploid products of meiosis are created in a sac called the ascus, plural asci, and each of the products is called an ascospore. By dissecting the sac, a researcher can isolate each meiotic product and place it in the culture dish to start a new yeast colony. Sexual reproduction in S. cerevisiae occurs when haploid cells of opposite mating type, denoted A and alpha, come together in an event referred to as mating to fuse and form a diploid cell, which then undergoes meiosis. S. cerevisiae reproduces both sexually and asexually. Asexual reproduction occurs by a process called budding, which involves a mitotic division of the haploid nucleus. After the division, one daughter nucleus moves into a small bud, or progeny cell. Eventually the bud is separated from the mother cell by cytokinesis. The yeast genome consists of 12 times 10 to the power 6 nucleotide pairs, and it is thought to comprise 6,268 genes. Its sequence was reported in 1996, the first eukaryotic genome to be analyzed completely. Paradoxically, some of these DNA molecules are smaller than the DNA molecule that makes up the E. coli chromosome. Yeast cells possess mitochondria, which also contain DNA molecules, although they are much smaller than the ones found in the nucleus, and they have a complex system of intracellular membranes. In addition, mutant strains with different growth characteristics can be readily isolated. The great differences between yeast and E. coli is the yeast is a eukaryote, Its 16 chromosomes are contained within a defined nucleus, and each chromosome consists of a single DNA molecule combined with a large number of protein molecules. Saccharomyces cerevisiae, baker's yeast. Baker's yeast came into genetics research about the same time as E. coli. However, long before it was commonplace in genetics laboratories, this organism was used in kitchens as a leavening agent for making bread. Yeast is a unicellular fungus, although under some conditions its cells divide to form long filaments which resemble the hyphae of other fungi. Like E. coli, yeast cells can be cultured on simple media in a laboratory, and large numbers of cells can be obtained from a single mother cell in just a few days. Obviously, research with such organisms can advance more rapidly than research with large organisms with a limited reproductive ability. E. coli cells and the viruses that infect them are tiny creatures that can be cultured in the laboratory to produce tens of billions of their own kind in a short period of time. These large population sizes allow researchers to screen efficiently for rare events such as the occurrence of a particular kind of mutation. Each type of bacteriophage, or phage for short, has a characteristic structure composed of proteins, each of which is encoded by a phage gene. The smallest bacteriophage have just a handful of genes, the largest have a couple of hundred. Because of their relative simplicity, bacteriophages are beautifully suited for studying basic genetic processes. Many important discoveries in molecular genetics have also come from studies with the viruses that naturally affect E. coli cells. These viruses, called bacteriophages from the Greek word meaning to eat bacteria, are much smaller than E. coli cells, about 1,000th of the volume. They attach to the cell surface and inject their DNA into the cell, whereupon the DNA is replicated and expressed to produce more viruses. As a result of these features, E. coli has become the microbial workhorse of geneticists. Many of the early discoveries in molecular genetics came from studies with E. coli. For example, the mechanism of DNA replication and the process of transcription and translation were elucidated by the work with this bacterium. Escherichia coli, a bacterium. The genetic analysis of bacteria began in earnest in the 1940s. Initially, several different species were studied, but in just a few years, most of the research became focused on Escherichia coli, a rod-shaped bacterium that occurs naturally in the intestines of animals such as ourselves. This organism can be cultured in the laboratory on a simple medium. It is amenable to all sorts of biochemical analysis, and mutant strains with different growth requirements can be isolated easily. Today, a select group of microorganisms, plants, and animals are favored in genetic research. These creatures, often called model organisms, lend themselves well to genetic analysis. For the most part, they are easily cultured in the laboratory. Their life cycles are relatively short, and they are genetically variable. In addition, through work over many years, geneticists have established large collections of mutant strains for these organisms. When gametes began, the organisms that were used for research were the ones that came to hand from the garden or the barnyard. Some early geneticists branched out to study inheritance in other types of creatures, moths and canaries, for examples, and as genetics progressed, research became focused on organisms that were well suited for controlled experimentation in laboratories or field plots.